When blood glucose drops, the liver can work to increase blood glucose levels. When blood glucose levels rise, the liver can respond by acting to decrease blood glucose. First, the response to an increase in blood glucose will be reviewed. Number 3. Insulin acts on the liver to decrease blood glucose levels. The liver regulates blood glucose after being acted upon by either insulin or glucagon. Insulin and glucagon are both secreted by the pancreas. Insulin regulates an increase in blood glucose. Two main factors regulate insulin secretion by the pancreas: glucose in the blood and autonomic signaling. Glucose in the blood is detected by beta cells of the pancreas to secrete insulin. The more glucose in the blood, the more insulin the pancreas secretes, and as blood glucose levels drop, insulin secretion from the pancreas decreases. Additionally, signaling from the autonomic nervous system can regulate insulin secretion. During exercise, when the sympathetic nervous system is activated, insulin secretion is blocked. At rest, and when anticipating a meal or just after having a meal, the parasympathetic nervous system can stimulate the secretion of insulin. Therefore, insulin secretion may be the result of nervous system signaling and not necessarily a result of the amount of glucose in the blood. Insulin is able to regulate blood glucose through GLUT4 receptors. GLUT4 receptors are the receptors by which glucose most commonly enters cells. When insulin binds the insulin receptor, it causes the translocation of GLUT4 receptors from the inside of the cell to the outside of the cell. This increases the number of available receptors for glucose to enter the cells. As glucose levels in the blood rise, and there is more glucose in the blood than in the cells, a concentration gradient is formed. This concentration gradient causes the diffusion of glucose out of the blood and into the cells through the GLUT4 receptors. Blood glucose levels decrease as a result. To review the role of insulin, insulin causes an increase in the amount of GLUT4 receptors on cell membranes to allow glucose to diffuse out of the blood and into the cells, which lowers blood glucose levels. In type 1 diabetes mellitus, the beta cells of the pancreas are destroyed and no insulin is secreted. Thus, glucose does not diffuse into cells through GLUT4 receptors. The glucose remains in the blood, resulting in severe hyperglycemia, and does not enter the cells, causing cell starvation. In type 2 diabetes mellitus, the beta cells are functional and there is plenty of insulin, but cells become less responsive to the insulin, leading to fewer GLUT4 receptors being available. This also results in hyperglycemia and the severe health problems associated with it. After a meal, glucose is continually entering the blood through the veins of the small intestines, causing blood glucose levels to rise. To review from our video on the hepatic portal circulation, all blood from the veins of the digestive tract pass through the liver before going on to the rest of the body. This means that as glucose enters the blood from the intestines, simultaneously insulin enters the blood from the pancreas. The glucose and insulin both flow through the liver first before reaching the other organs of the body. The insulin allows the liver to increase its GLUT4 receptors and take in up to 1/3 of the newly absorbed glucose. The liver is then able to store this glucose as glycogen. Glycogen is a chain of glucose molecules kept inside cells of the liver as a storage place for glucose. The liver can also convert some glucose to fat, which the liver then secretes to be taken up by adipose tissue. Insulin can also increase GLUT4 receptors on skeletal muscle, and skeletal muscle can form its own glycogen. However, during exercise, when insulin levels are low, skeletal muscle can induce GLUT4 translocation by contracting. Thus, skeletal muscle does not require insulin to take in glucose during exercise. Additionally, the brain and kidneys never require insulin to take in glucose from the blood.